And our final speaker is Stephan Pless from University of Copenhagen. Stephan actually is combining a lot of different techniques in his lab, studying the function of ion channels, and basically he's trying to answer 3 different questions, if I understand it correctly from the profile of your lab, is how small molecules and peptides bind to ion channels, then how protein-protein interactions or mutations alter the function of ion channels, and how conformational changes also influence function of ion channels. Looking forward to it. Thanks.
Thank you. Thank you very much for this kind introduction. So thank you very much for having me here to talk about something that isn't quite included in the list of the things that we are really interested in my group. So, and that is sort of trying to leverage some of these new technologies to make new binders against targets that we know. And in this particular case, we're interested in ion channels. So the agenda is basically, I'm going to tell you about a project that is a bit further matured, where we've tried to make binders against an acid sensing ion channel. And then the second part is sort of more of a play project where we try to make binders against one of our other favorite proteins, the sodium leak channel. So to start with the first part of the talk, I really have to give credit to Janina Zöllmann. Some of you know her. She's really the star of the show here. She's really taken on this project and run with it. I really have to give her credit for running every single aspect of the project. It was Sam who originally had the foresight and vision to actually implement this computational approach in the lab, but it's really Janina who took it on for this particular project. And there's a few collaborators to mention here. Joe Rogers is a colleague of ours, and the swank-looking student is a postdoc in this group right now. And then there's Søren, who is also my voice control man here for this talk, who has helped us a lot with this. So acid-sensing ion channels, just very briefly, they are trimeric ligand-gated ion channels, but they are gated by the smallest envisionable ligand, which is protons, basically. So when protons bind, they open. They're probably involved in synaptic plasticity, but they play mostly a role in pathophysiology, including in pain, cancer, and stroke, and I'll get back to that. So we're interested in these channels quite a bit in my laboratory, and part of it is because we're interested in the pharmacology of these. And it is actually a rich pharmacology. So everything you're about to see, you could call a waste of time. It's sort of a proof of principle that we're going to try to establish here. So there are small molecules. As is often the case with small molecules, they're not terribly selective, so they're not often very useful. But there is a rich pharmacology in terms of venom-derived peptide toxins that target acid-sensing ion channels. Some of these are actually quite potent, but none of them are truly subtype selective, and often they are very stable, as Michelle nicely pointed out. So they're very powerful tools, but they're also not easy to make in many cases. And so we wanted to, with this project, we wanted to sort of get into the question, can we make easy to produce subtype selective modulators? Because one of the other things that you have to consider here is we have these nice venom-derived peptide toxins, but actually nature has decided where they bind. It's not you deciding for a particular region in the protein that would allow you to make something subtype selective. So that's what we were trying to get around. And we're basing everything that I'm going to talk to you about on the work of David Baker, who got a Nobel Prize for this last year, basically. So it's a computational approach called RFdiffusion. So the idea is this. You have a protein of interest. Let's say this is our ion channel, the blue blob here. You need a high-resolution structure. And then you ask the computer, make something that is shape complementary. It doesn't involve any chemistry at this point, just backbones. What would be complementary to the shape of the target, basically. And they've used this in beautiful work, actually, especially if you're interested in this kind of work. I would really recommend looking at this here, where they basically use binders to neutralize snake venom, which is a really cool story. Now, in our case, what we do is, in the first step, we use sort of the computer to make backbones, indicated here. We fit them then with amino acids, and then we ask AlphaFold, is this actually a good binder? Okay. Then if AlphaFold says yes, gives a thumbs up, then we go and do high throughput expression, often in 96-well formats. We then use SynchroPatch or similar high throughput methods to screen these. And then sometimes we also go more into the biophysics or even animal models. The beauty of the system is that all of this, I'm going to exclude this part, but all of the 4 on the left here, they take about, I would say, 2 to 4 months, basically. So it's really quite rapid overall. So we started with the ASIC-1A structure. In this particular case, we actually defined a so-called hotspot. So you can let the computer run wild, or you can tell the computer, I want to go after these residues. In this particular case, we went after these residues because we knew that one of the more subtype selective binders, in this case Membelgin, is actually binding there. And we thought if we want to have a chance to get something subtype selective, we'll target that part of the channel. So that's where we wanted the computer to target something. And we essentially told the computer, make backbones that range from 40 amino acids to 100 amino acids and just run with it. The computer essentially does what you can see here. So it runs sort of in this computation, and you'll see the peptide eventually finding its place where it wants to be. And we can do this with many of these. So in this particular case, we use 12,000 backbones. And each of these backbones, we fitted with 6 different sets of amino acids. So you have the backbone in place, and then you tell the computer six different combinations of amino acids. Optimized for stability, optimized for solubility. So you end up with 70,000, which is something that's even beyond the powerful capabilities of SynchroPatch. So we have to boil it down, and that's what we use, basically AlphaFold. So we use, in this case, PAE scores. There's debate about whether this is the best scoring mechanism. That's the one we've been using, but there are other ways to score your peptides, basically. In this case, we went with 77 binders that we expressed. Briefly, we clone them using Golden Gate. It's very easy. We transform them, express them in 96 well plates. We then purify them in 96 well plates because they actually have a his tag attached to it that we can then cleave off. And then we can literally elute it in a ananeon buffer, essentially. So then we can go directly on the SynchroPatch. So it's a very efficient, very rapid workflow. This is completely blind. We don't even check if a particular protein is expressed. We just go for all of it, basically. And then we go straight on the SynchroPatch. The protocol is this. We activate the channels here with a pulse of low pH, and then we apply the binder and check again. And what we were actually looking for inhibitors in this particular assay. So this is what it looks like. This is your plate. We're using actually hex cells because as some people in the audience found out a long time ago, hex cells actually have a beautiful ASIC-1A endogenous current that you can use for this purpose. And so, you know, predictably, in most cases, it looks like this. You have your control current shown in black, the one with the miniprotein that I call A1 here in green, and it looks exactly the same. So in other words, the miniprotein binder doesn't modulate the channel, but in some cases, they actually did. And in this case, I'm showing you miniprotein G7, which is one that we've worked with the most. It actually inhibits the channel with around 20 nanomolar potency. And this was really, I really cannot stress this enough. This was the first time we ever did this in the lab. And I have zero computational background. Yeah, I went to a Waldorfschule for the Germans in the audience. Okay, I grew up without a TV or computer, so it's really the postdocs that really established this in a relatively short time frame and managed to get this to work. So 2 out of the 77 that we tested actually inhibited. And the beauty of this workflow is you can actually now go back. You can, once you have binders here, you can go back to step 1 and say, start with what we know works and optimize on that. So the way we do it is, I told you that we typically just fit 6 different sets, but you can tell the computer fit more sets of amino acids to a successful binder or like a modulator. You can alter the temperature parameter, which basically means you have more noise in the system and you get more variability in the backbone structures. And then finally, you can do something that's called partial diffusion, which I love watching. Oh, I want to show you this video. It's really funky, actually. So you keep part of the backbone in place and you only vary the rest. So you can see this here. This part of the backbone was in place and you only vary the rest. And you can do this. And we call this optimization of the validated proteins. And you use the same protocol, basically. And this is a plate. And if you just want to focus on this below, this is before we apply the miniprotein. And then we apply the miniprotein and the current almost go away, basically. Okay. So here we actually ended up with many inhibitors. 25 out of the 72 new designs inhibited, but none were actually better than what we had before. So all the IC50s were actually slightly higher than what we got before. Obviously, this doesn't mean that this doesn't work, this approach. We can iterate on this more times and using different parameters. This was just a first try. In other labs, I should point out the David Baker lab, they have had much more success with this optimization. We haven't been awfully successful with it as of yet. But we had a binder that works really well. It's about a 40 amino acid peptide or protein, as depending on how you define these things. And we wanted to use it in a stroke model. So this is the work done in the lab of Chang-Si Kai at the University of Copenhagen. And I should say I'm not an expert in this. So what they do is they basically induce a stroke in the mouse, and then they use reperfusion. Then we do a relatively brutal method where we directly use intrathecal injection of these peptides of either a positive control or just buffer or a peptide, obviously. Then we extract the brain, we cut it, and we stain it, basically. And so what will be shown in white is the part of the tissue that dies. And when you have a peptide there that rescues the cell death by inhibiting ASIC, you will see it as normal color, typically red, basically. And so I really want to make this very clear that what I'm about to show you is very preliminary, and we will unblind part of a randomized study that we have going on right now. So what I'm showing you is very preliminary data that we just got from a few animals. So on Monday, we will unblind, and you'll find me crying in my office and going back on and apologizing to all of you for showing you this preliminary data because maybe it's wrong. So basically, when we treat it with buffer, it's kind of a bit hard to see here, but you see there on the right side here, that's where the stroke is. So you have dead tissue, basically. And in the very few animals that we tested beforehand with G7, it was mostly gone, basically. So this looked very promising, which is why we now sacrificing a lot of mice going into like a randomized study, essentially, and double-blinded. But I don't know the outcome. So I'll know on Monday of the first 40 animals or something like that. At which point we might stop if it doesn't look promising, because it's very expensive and very time-consuming. So it looks good, but we'll see how it actually holds up. So in conclusion, we've shown that we can design and functionally validate these proteins. We've also, and this is important because ASIC pharmacology is notoriously tricky. It can change depending on the expression system slightly, and we've also validated that at least our G7 binder actually works very well also in other expression systems like xenophosphate oocytes. And we have these stroke models going on. We haven't done much in the way of biophysical characterization. Probably these binders, unlike the ones that Michelle was talking about, they're not very stable. Okay, so this is not something that you can give to a patient anytime soon. Okay, it's just a starting point. It's a proof of principle, if you like. And we know that the peptide that we're most interested in has extremely high selectivity over something like ASIC3, but we are now testing all the other ASIC subtypes. We haven't done those yet. Good. Second part of the talk that's going to be a bit shorter is basically what I call breaking up the party. And it's targeting the sodium leak channel with de novo binders. Now, this project was initiated by actually a PhD student in the lab, but she ran into some issues, and then Sam, with his computation pipeline, said, why don't we clean up, and I'll try to help you out with these de novo binders to get to the question of your project. Now, before I go into the details, I have to tell you that there is this sodium leak channel complex that some of you may know, some of you might not. So I'm just going to very briefly introduce it. So it's a sodium leak channel. Genetically speaking, it's very similar to voltage-gated sodium and calcium channels, except it is functionally very different. First of all, it's a leak channel, and it doesn't act itself. So if you just express the channel part, which is this, you will get no current at all. You need an auxiliary protein called FAM155A and two other auxiliary proteins, UNC79 and UNC80. I should mention that UNC80 and UNC79, they're 2.5 and 3,500 amino acids. Okay, they're huge proteins. You can't really appreciate it here. The whole thing is about 300 angstrom wide. So it's a massive protein complex. And remember, you need all three to get function. So this looks like if you just put NALCN in a cell, nothing happens. You need all four proteins to get any kind of function, okay? And this will become important later on. Why are we interested in this? It's a widely expressed channel. We don't know exactly where it's located on a subcellular level yet. We do know that loss of and gain of function cause very severe neurodevelopmental disorders. And there's no pharmacology, literally zero pharmacology. The most potent inhibitor of this channel to this date is calcium. Okay, that's absolutely useless. You can also use gadolinium, but that blocks just about everything else as well. So it's not very good. And there's also no good antibodies. There are claims now that there is an antibody, but I haven't seen any data, so I don't know. I don't know yet. So basically, we wanted to see, I should say, the first part that we tried is make mini binders against the outside. We failed completely. Okay, we have failed completely. We can talk about, if you are interested, we can speculate on why that is. What we were wondering is, can we disrupt protein-protein interactions on the inside by targeting specific interactions between these UNC proteins in the channel? So this is the side view again. We know that part of the channel that we call the D2, D3 linker touches onto these UNC proteins, and we were wondering if we can just make a binder that competes with that. And we knew that this could work because we knew that if you just express all of this together with just the linker, the linker will displace the UNC proteins and kill the function of the channel. Okay, so this will be a control later on. We can express all of this together and then just express this on the side, and this will interact with these UNC proteins and dislodge the UNCs and render the channel non-functional. The other contact point is actually an interesting one. It's a smaller loop, D1, D2, and there's a tiny loop here, and there's a tryptophan. That's all important. So this is a 1 million Dalton protein complex. If you mutate this tryptophan to even something like a phenylalanine, the whole thing falls apart. Okay, so there's a lot of importance in this loop. And we were wondering if we can essentially displace this loop also with a mini binder. So we had 2 contact points here that we wanted to target with mini binders. We're using the same kind of idea. We use RFdiffusion to make backbones, in this case 5000. We use ProteinMPNN to make sequences, to fit amino acid sequences basically. And then we use AlphaFold to select the supposedly good ones. In this case, actually, we were spoiled for choice, because it turns out if there's a naturally occurring protein-protein interactions, the computer has a very easy time to actually generate binders against these interfaces. So we actually ended up with 400 predicted binders. We only decided to go ahead with 40 of them. We made sort of an initial brief screen, like a very crude screening. We followed this up, and I'm just going to show you some of the data here. So this is the control here, NALCN itself. So this is the control. We end up at 1. This is a normalized current, essentially. Now, this D2, D3 linker that I showed you before, you can knock down the current to about 20 to 30%. Most of the binders didn't do very much, but one actually was as potent as our positive control, basically. So this binder we call D3. And it actually is predicted to interact with the D1, D2 linker that I mentioned just now. So, or it's predicted to compete with it, I should say. So this is the structure of the binder. I can show you the structure because our IP office had no interest in this. So supposedly, based on the computational analysis, these are the most important residues, as is this tyrosine here, potentially also this one, but not this leucine. So we wanted, can we verify that the computer predictions are actually correct? So we basically have our outward current here plotted, NALCN itself, with the linker, with the D3 binder, so you knock the current down. If you mutate either of these residues here individually or together, you lose the effect almost completely. And this is where it gets interesting. This phenylalanine here overlays perfectly in structure with the tryptophan that I was telling you about. So we're wondering what happens if we put the tryptophan there in terms of the phenylalanine, and we get our inhibition back, basically. It gets a bit more tricky with the tyrosines. So if we mutate this tyrosine, the effect is gone. If we mutate this tyrosine, we get sort of an intermediate effect. And if we mutate both of them, actually, it's a pretty good binder. We're not quite sure why that is. We think that maybe by removing the bulk here and here, these phenylalanines are able to wiggle into their binding pocket deeper, but we don't know. We haven't tested that in more detail. But as predicted, this leucine here up here doesn't have a big effect on the inhibition basis. We also wanted to see, what's really curious is that actually most of the patients that are known, they suffer from gain-of-function variants. And we wanted to see if these gain-of-function variants are also sensitive towards inhibition by these kind of artificial binders. And this is the control experiment. This is NALCN wild type, this is NALCN alone. With the linker or our artificial binder, we get a good reduction. And the same thing happens if we now use an R1181Q patient variant, which is a massive gain of function, but it's normalized here. And it's inhibited by both of these variants, also, by these peptides or peptide mini-protein binders, basically. So although this is a massive gain of function, they are still sensitive to these kind of peptides or artificial protein binders. Good. In conclusion, I've shown you the design and validation of a mini-protein capable of intracellular binding and competing with intracellular protein-protein interactions. And we've also started to mutationally verify this interaction. And it seems to be quite potent because it inhibits even strong gain-of-function variants. The problem is we cannot really get to sort of KD values or something because we cannot actually express these two proteins in isolation and do, let's say, I don't know, ITC experiments or something like this for to get a proper KD, simply because these proteins are way too big and you cannot chop them down into smaller pieces. And believe me, we've tried a lot. Next up, we're going to try the inhibition in neuronal cultures. And yeah, again, we've tried a lot. We haven't succeeded on the outside yet. We're still trying, but we haven't so far. I don't want to oversell these mini proteins, so there are challenges with them. You need a high resolution target. You ideally need a stable cell line to also do the screening, basically. If you have shallow or hydrophilic surfaces, very, very bad. So all of the NAVs and CAVs are terrible targets. PAE scores are quite good, but not always ideal, so there might be better scores out there. And in vivo stability, immunogenicity is still out there. We don't know. We just have to test, and we probably have to optimize a lot. but it's really, really moving very fast. So the Manu Benjoni group just published a very nice paper in Cell where they've used intracellular binders using a more simple approach against sodium channels. And the Baker group put out a preprint on GPCRs, very beautiful work by others there that moves the field forward quite a bit. Good. So I've talked to you about the design of new binders against both acid-sensing ion channels, against this sodium leak channel. And with that, I would like to sort of finish off, thank my group. I've mentioned the people that are relevant to this project. Our funding, obviously, we're very grateful here. Our environmental program that led to the fact that I came here by train all the way from Copenhagen. And then I want to point out that yesterday we finally got the okay from EMBO that EMBO will actually sponsor the next iteration of the EMBO FABS lecture course on ion channels and transporters in Eritrea in May. So for all the trainees, it's only for trainees. Keep this date in mind, May 13 to 19. The speakers are listed on the left and right. This is what it's supposed to look like. I've never personally seen it like this, but the last time the weather was terrible and everybody got COVID. I hope that none of these things will happen again. Otherwise, it's a beautiful course. Please consider joining once we have put it up on the website. And with that, I'm happy to take any questions.
Thank you. Very nice talk. I'm just wondering, you said in the end, hydrophilic surfaces are a problem. So this is not a good way to maybe design pore blockers, for instance? Just that would be...
Yes and no. So basically, the current state of the art is that these computer programs, they're designed to go after hydrophobic patches, which is kind of rare on extracellular surfaces of like a ligand-gated ion channel, for example. You're absolutely right that if you make pore blockers, you can kind of sometimes, you can actually, so we're doing this on a different protein. You could guess it's a potassium channel where there's many pore blockers that actually have a lysine sticking in. The computer will come up with this solution also. It's just the scaffold around it will be slightly different. But yes, pore blockers are absolutely it's absolutely possible. The problem is that the pore blockers are often not terribly selective unless you stitch on a bigger framework that then makes it selective because of other residues that are subtype selective. Does that answer your question?
Yeah. And during this diffusion process, do you vary the sequence of your mini proteins, or do you always have one sequence and you're sort of optimizing the position only?
So this is a good point. So we're using, we started to do this a year ago, and we are already in the field. We're considered completely outdated because now you can use Bindcraft where everything is done in one step. Both the backbone and the amino acid fitting is done in one step. What we're doing is first, we're actually making a backbone, and it's just the RFdiffusion is just doing the backbone. And then it's the second step that ProteinMPNN fits it with amino acids. So it's a two-step process where you start with a backbone and then you go in with the sequences. The newer versions like Bindcraft, they do everything in one step, and there you have a bit more, you need a lot more computational power. It takes longer, but your output is more likely to be an actual binder or modulator.
Thank you.
Thanks, Stephan. So do I take it, we've just heard about the natural toxins, obviously have disulfide bridges, are very stable. You don't have that in your proteins then by design, but can you incorporate that?
We could. We actually tell the computer not to. Okay. So we ask the computer no cysteines, no methionines, nothing that would be chemically reactive or tricky during synthesis. We want to keep these as simple as possible, basically. So this is by design. You can go down the route and actually tell the computer, put in disulfides and try to do it this way. But there are complications with, like the beauty of these peptides is they express like crazy. We're expressing them in 96-well format, do a very crude purification and throw them in a synchro patch and they work immediately, basically. So that's the advantage, but there is disadvantages in terms of stability and things. We will have to cyclize or come up with other things to make them more stable, if that makes sense.
Yeah, so when you're wondering whether your stroke efficacy is there or not in that study you mentioned, do you know how long that peptide is going to stay around for?
No, we're doing it in parallel with the mouse work. We're a bit under time pressure now.
Yeah, good luck.
Yeah, thank you. We'll need it.
Very cool project and great presentation. I was wondering if you see also maybe the possibility to design these mini proteins against synaptic plasticity related receptors in the brain. So for example, NMDA receptors that might be good for neuropsychiatric diseases later on.
Yeah, I think the race is very much on. I think many groups are on this. I don't know if in if there's any group working on this in particular, but I think we'll see a lot more of this in the future. Also, you know, if you move away from receptors, you actually have an easier time. If you have a small soluble protein, then you often it's easier to make binders against those. So you can indirectly affect protein-protein interactions by targeting not the receptor, but maybe the binding partner.
Nice talk, thank you very much. Have you looked for binders that don't have any functional effects? Because that could be an interesting niche.
Yes, no, you're absolutely right. And sorry, I should have made that more clear. So our assay is only designed to pick up modulators. So there is a good chance, and I keep joking about this, we could be missing out on femtomolar binders here. We don't know. If they're just binding and have no functional effect, we'll absolutely miss out on them. It's just a question of the assay, basically. So it's basically what you want to go after. So there are other assays where you could screen for that, but we could be missing out big time here, which is a bit depressing.